Hi, I'm Darren Woodside, Vice President for Research here at the Texas Heart Institute, and we're here in studio today talking with Dr. Anthony Farrelly from the Indiana School of Medicine about his Grand Rounds presentation today, which was talking about how certain proteins in the heart are involved in generation of the heart during embryogenesis. Welcome, Dr. Farrelly. And
thank you very much for the kind welcome and the wonderful introduction. As we talked about earlier, I lived in Houston for a couple of years, and it's very nice to be back what feels like home to me.
Especially now when we can travel and meet people again, right?
I know. From the long, desperate of travel from COVID, it's very nice to see that, when you get to see like new people, young scientists, it's very invigorating, and I kind of missed that over the last 2.5 years.
That's true. Well, part of the beauty of science is being able to express it to other people and share it.
And like learn new things from people and get good ideas, and that's the fun part of the job,
right.
For sure.
Well, I learned a lot from your Grand Rounds today, and a lot of times it's very difficult to put organ generation in perspective, and it's difficult for a lay audience to kind of follow that. But I thought you did a really good job in describing the fundamentals of heart development.
Thank you.
One of the things that you said that struck me was, and I think it'll resonate, is that as the heart develops, it's just a tube that kind of folds over itself and septates. I'm wondering if you could just elaborate a little bit on that for maybe a general lay audience.
Sure. So when you think about how we all start as a single fertilized cell, and then one cell goes to 2 cells, 2 go to 4, 4 go to 8, 8 go to 16, 32, etc. At some point, you just have to stop becoming a giant ball, and you have to start deciding as an embryo what's going to happen. What's going to be the head, what's going to be the feet, anterior, or your belly or your back, your right side, your left side. And this is orchestrated through a very complex process that involves a lot of signaling and a lot of transcription regulation, where there are proteins that bind to DNA and coax that DNA to either turn on or turn off. And so when you get to the point of heart development, the heart starts as this kind of flat sheet of mesoderm mostly, and it kind of rolls into an upside down smile. We would call the cardiac crescent, but it looks like a frown. And then they move towards the midline and they form a tube. And so that tube is going to be an outer layer of muscle and an inner layer of endothelial cells, which are like the sleeve inside every vessel you have in your body. So new cells come in from the top of the tube and the bottom of the tube, and they do it kind of asymmetrically, so the tube starts to bend. And where you have like your atria, which are at the top of your heart, they end up being at the bottom of the tube, and your ventricles, which are going to be at the bottom of the heart, start at the top. And so what happens is that the atria then loop over the ventricles, so they become on the top and they become on the bottom. It kind of looks like a big, kind of C.
Hence the tube folding over on itself.
Yeah, folding over on itself. And so then what happens in both the atria and specifically in the ventricle, a group of cells in the middle of that tube or that C recognize that they need to proliferate and grow up. And so they cut the tube in half, where one ends up being the right ventricle and the other one becomes the left ventricle.
And that's septation.
And that's septation. And that's a process that's required because in mammals and in birds, we have separate circulation where we have deoxygenated blood that stays on the right circuit of the heart. So the right ventricle pumps blood to the pulmonary artery, which takes it to the lungs to get oxygen, and that comes back to the left atria, gets drained into the left ventricle and pumped out to the body. And that happens billions of times in a lifetime. And so when that process of forming that very elegant structure goes a little bit wrong, you get defects in the efficiency of the pump, and that can lead to serious conditions that require, in many cases, immediate surgical intervention, where like people at the hospital here encounter every day. And so what I have always been interested in for many years is understanding the stereo instructions, the molecular pathways that make that heart form normally, so that we can try to understand when we see and encounter defects in that process, which occur almost 1 in 100 live births. So it's the most encountered congenital defect to have a heart abnormality. Most of us know someone who has a cardiac defect. Whether they know it or not, you're probably talking to somebody almost every day.
And that kind of plays into the title of your talk, right? Building a heart hand by hand. I was wondering if, for a general audience, you could describe what hand proteins are.
Yeah, so I'm really good with puns, I guess. And so there are a family of transcription factors that have a particular secondary structure. So if I was talking to a lay audience, have you ever seen a number of different kinds of hammers? Have you ever seen a number of different kinds of screwdrivers? Some have starry ends, some have flat ends, some are long-handled, some are short-handled, but they're all screwdrivers. So basic helix loop helix transcription factor genes code for proteins that have a particular shape that allows them to interact with other BHLH proteins and bind DNA. And so the hand 1 and hand 2 that I've been studying now for many years, they play roles in a number of different organ system development, including the heart. And so by using that as an entry point into these pathways, I try to figure out what is regulating those genes in the process and what my factors are regulating downstream in the process, so that we can put a comprehensive picture together of this one sub-pathway and hope to link it to other pathways from other factors, so that we can get this network of genes that fire at the right place in the right time to make all of these different cell types behave and form a normal organ.
That's really complex biology, right?
It's really complex to think about, which is why we're just focusing in on a tiny little piece of it, and hopefully will give us some insight as to what other pieces we should look at next once we kind of sort this process out.
One thing we really like to do here at the Texas Heart Institute is translate a lot of our basic research discoveries into things that can eventually help patients. I was wondering if you had some thoughts on how you foresee some of the basic research that you're working on, how that might be targeted to maybe new therapies in the future, or how we can improve patients' lives with it.
Sure. So one of the key mechanistic regulatory points of BHLH proteins is they have to work in pairs. And so how these proteins interact with each other is a regulated process. So you can have two of the same protein form what's called a homodimer because each side's the same. And then you can have one of these proteins interacting with a completely different kind of BHLH protein, and that's a heterodimer. And that regulated process, if it is being disrupted in any of these abnormalities that we're encountering, could be mitigated by using a series of triggers or drugs, if you could find a small molecule that control that at critical times, to make the bad interactions not happen and the good interactions actually occur. The oil or the fly in the ointment here is you have to know where the timing is critical for the event that's deleterious to occur. And you need to know what the post-translational state of those proteins is at that time in order to have a window of what to target. And the challenge is just getting more information to understand how this process occurs over time in a better way.
And that's really the most important part of basic research and discovery science,
is
building the tools and providing the targets that we can then look at to try to develop better therapies for patients.
Absolutely. Yeah. And, the search through the human genome for variations in sequences that are within hand 1 and hand 2 and upstream and downstream of hand 2 has identified a number of abnormalities that can be associated with diseases. And so we're excited about the possibility, as well as being able to find, how the proteins work if we can understand variants that don't do their work so well, or we can find variants that control how much or where a protein is expressed, you could potentially mitigate those with genome editing at some point in the future, where you could correct the change and then, understand that you get normal expression at that point and avoid the defect that would ensue.
Well, thank you very much for the work that you do, and thank you for coming to Texas Heart Institute and sharing that with us today.
It's
for
been my
Grand
pleasure.
Rounds.
This has been a wonderful experience, and we'd like to say thank you again for giving me an opportunity to share my work.
Very good. Thank you.